Good afternoon, or good morning, or good evening, depending where you are. We're starting short three-part series about basics of cytology. In order to make these three sections a little bit easier to handle, I decided to divide them into three parts. In the first part, we're going to take a look at the cell in general, but paying most of the attention to its plasma membrane, the cytoplasmic membrane. Then in the second part, we're going to briefly address the most important organelles. And the third part will be about the nucleus and the DNA, cellular division, DNA replication, and also protein synthesis on the basis of DNA. So just to let you know that this section is going to be just mostly about cytoplasmic membrane. So here we have one generalized look at the cell, nicely illustrated to show all of the important cellular structures, but of course such a cell doesn't really exist within a human body. However, it addresses a number of different things such as plasma membrane, but as we can see on one part of the cell illustrator, added some microvilli and also added a couple of cilia that would make eventually some cells being capable of moving. On a cross-section through the cell, we can see here the centriole, we can see the mitochondrion, we can see multiple lysozymes, secretory vesicles, Golgi complex, endoplasmic reticulum, and of course, what is really important, nucleus itself with its own nuclear membrane and somewhat kind of granular looking cytoplasm, nuclear content, which we know is practically the DNA. Anyway, so let's move away from this idea of let's try to see the cell in everything that one cell should have by focusing into this segment, which is one of the most important to accept to understand what's going on. It is cytoplasmic membrane or the cellular membrane. In a previous section about organic chemistry and some compounds that we had to address, we talked about phospholipid molecules that form bilayer of phospholipid, practically making itself the most important structure to assemble the cellular membrane. Phosphate groups that are polar heads, that are hydrophilic, are oriented both outwards as well as inwards into the cell. So their interaction with molecules of water that are coming in abundance in both cytoplasm as well as immediate cellular surroundings, extracellular fluid compartment, is going to be perfectly well coordinated. Fatty acids that form hydrophobic tails are oriented internally, so they're making the core of the membrane. And if you take a look throughout this picture, you're going to see that phospholipid bilayer certainly dominates. However, it's not the only ingredient that we need to add to have a good quality understanding of what cytoplasmic membrane or plasma membrane is. We can see here a variety of different molecules that are attached to the membrane itself, or they're allowed to completely penetrate and perforate the membrane, such as example of these three integral transmembrane proteins. Some of them would be interesting from the point of view that this cell becomes recognized as a part of organism, therefore immune system will not respond by challenging the cell or recognizing it as a foreign tissue. Some of the proteins, like this one in the middle, is obviously illustrated and properly labeled as a channel, something that will enable quicker and easier transport across the cellular membrane through its central opening, which is called the pore. There are additional molecules that appear to be attached to either outside or inside aspect of a cellular membrane. They are either glycolipid molecules. Of course, glyco means containing sugar, lipid attached to it, glycolipid, or glycoprotein molecules where the protein would have addition of some carbohydrate part. These these proteins essentially are there to fulfill other important functions, But we need to think about cellular membrane, its bilayer of phospholipid, and we're going to pay specific attention to transport across the cellular membrane. So this is pretty much the introductory part of why we need to understand. As we know, phospholipid bilayer, as illustrated on 6 different segments of this screen, would have hydrophilic and hydrophobic parts of the molecule. Because the water surrounds cell as well as fills it up from the inside, it would be very easy to understand why it's good to have phospholipid membrane molecules with its phosphate group oriented towards water-filled compartments. Water will not be repelled, rejected, but water doesn't have an easy pass through the phospholipid bilayer. Neither would be easy for ions, regardless of their minuscule size and their charge, to just keep passing in or out of the cell as they wish. Therefore, the plasma membrane really maintains the homeostasis of the cell itself, making sure that its internalized content is fully compatible, or better to say, optimized for proper functioning of the cell, and also that any compounds that are coming in a surplus within a cell essentially could become exported and therefore not allowed to disrupt the homeostasis. On the other side, cell cannot be completely turning its blind eye or blind ear to what's happening in its immediate surroundings in the extracellular compartment. Cell would have a need that from time to time allows extracellular compartment to stimulate it in order to do certain function. And I don't want to speak always in these general terms, but for example, what is required for a muscle cell to cause contraction? It needs electrical stimulus, a nerve impulse, which has to arrive to the surface of the muscle cell, which then triggers a set of different preferred responses that would lead to muscle cell contraction. Or cell sometimes needs to respond by secreting some of its content, which has been manufactured within a cell. And it is not done randomly or just when the cell completes its task. Basically, it has to be done with the aim of maintaining the homeostasis of the entire body. So as we can see here, different proteins that are integrated through the phospholipid bilayer could serve either as ion channels or transporters or receptors or eventually enzymes or cell identity markers, or simply being used as some kind of linkers that will allow attachment of different other organic molecules or organelles directly onto the surface of a cell. I would not prefer that we start our discussion with this diagram. I would go to the next slide just to try to hypothetically explain this and to help you understand what's going on. Although the cellular membrane is exposed into 2 different water-filled environments, this light blue color is for extracellular, beige, light beige is intracellular fluid compartment or cytosol. Same applies to lower diagram. So we're talking about 2 different gradients that the cellular membrane must maintain. One is concentration gradient, which comes from a different distribution of different ions on 2 different sides of the cellular membrane. So, for example, a sodium is known to be the major ion of the extracellular compartment, whereas potassium, illustrated in form of these little triangles, is the most important and most abundant ion within the intracellular compartment. And as you can see, both sodium and potassium carry positive charges, and it's the role of cytoplasmic membrane to keep abundance of sodium outside of the cell and abundance of potassium inside of the cell. Even on this diagram, although it is very generic, you can see only this triangular structure representing a single potassium ion in the extracellular fluid. Obviously, to keep more on one side and less on the other side of the membrane has been successfully done. If we reverse and try to understand what's happening to sodium, we're going to see only one green bubble representing sodium ion on the inner aspect of the cell within the cytosol or intracellular fluid, where majority of potassium, sodium ions is held within the extracellular fluid. Oxygen, naturally, that the cell would need for its own metabolism, appears to be part of this diagram, more present within extracellular fluid compartment, and it will pass into the cell, which will consume it for its own metabolical purposes. Carbon dioxide that would be produced as the end result of metabolic cycles will be more concentrated within the cell, but will then gradually be allowed to diffuse out of the cell and to be eliminated as a byproduct of our metabolism. So I believe that concentration gradient is always fairly easy to understand what's going on. Electrical gradient might be a little bit more difficult to grasp, but I hope we would be able to do it together. The total sum of all charged particles in the extracellular fluid compartment is not the same as the total sum of all charged particles within the intracellular compartment. Regardless of their absolute charges, whether they're positively charged or negatively charged ions. I'm talking about ions in general and the sum of all charged particles. So when we would compare extracellular fluid versus intracellular fluid, top versus bottom of the lower diagram on the screen, it'll turn out that there are more electrically charged particles on one side of the membrane than on the other. That's why the illustrator doesn't have other choice than to put plus signs where more charged particles are found, extracellular fluid compartment, and negative sign for intracellular fluid compartment where we have less charged particles. Negative sign doesn't only reflect, oh, it must be more of a negatively charged ions. No, quite the opposite. It's not negatively charged particles. It's the sum of all charged particles being unevenly distributed across two surfaces of membrane. So instead of seeing it like negative positive, we can think about more or less positive or more or less negative. We immediately understand the difference when we say something is good, but something is better. We understand the difference, something is charged and something is more charged. So I'm asking you to try to remember that we're talking about just different aspect of charged particles on both sides of the cellular membrane. And that is also one of the gradients that cytoplasmic membrane must maintain. Now, with hopes that this part has been properly accepted and understood, we can go to the previous slide and come back to understand the role of different proteins that are integrated into cellular membrane or plasma membrane, and they both would have openings on both sides, extracellular and intracellular fluid. So let's see here how it works when the integrated protein is acting as the ion channel, upper left corner of this slide. So what we have is that ion channel doesn't work as the generic channel for all ions. It is specific. So, for example, this particular protein serves as a channel only for particles of, let's say, sodium, which we know, more concentration in ECF, extra, less concentrated in intracellular fluid, cytoplasm. So we would need to open up this protein and to allow that just simple concentration gradient to push sodium into the cell by diffusion, going from higher to lower concentration gradient. When the protein would close its pore, the transport must stop. Therefore, all across the cytoplasmic membrane, we're going to be seeing some protein channels that will allow only transport of potassium. Some of them will be only for transport of chlorine, negatively charged ions, or phosphate ions, or bicarbonate ions. So that's why we say that ion channel is specific for just one type of ion that it would allow transportation. All right, so we have here a similar concept, but this time we're talking about protein being called the transporter. This one is perhaps a little bit more challenging to understand how it works. Obviously, the size of a transported substance is far greater than the protein itself or its opening the pore. So what's happening here is that transporter protein shortly or temporarily binds with a substance that needs to be transported. And then, as it goes through conformational change, basically the transported substance becomes sucked into the protein, which releases it on the other side of the cellular membrane. If this is too difficult to accept as a concept, maybe you can think about conformational change as a glove, which when we properly put on our hand, it has inside and the outside, and we just kind of peel it off our hand. Inside turns out, inside becomes in. The same glove, just going through conformational change, and that is the essence of work that transporter proteins would do. Some proteins will act as receptors, and there is no transport this time. Receptor just means one thing. On its either external or internal surface, the protein will have the binding site. And it will be universal only for one specific type of molecule that can bind to the receptor. That molecule that binds to a specific receptor, we give generic name, we just call them ligands. So whatever ligand is, basically it cannot bind to any of the receptor proteins. It has to seek and make connection with specifically created receptors. You can see here that example illustrates antidiuretic hormone, that is the ligand, which binds to receptors that kidney cells are having. And as a result of this hormone's activity on the kidney cells, it'll change the water permeability of certain plasma membranes that would reduce amount of water that would be excreted through urination. So that's antidiuretic hormone, making membrane with its presence just a little bit more tight and preventing unwanted losses of water. So ion channels, transporters, or receptors. Some proteins, as we know, could be exposed and act as enzymes. We know what's going to happen. They will bind substrate, help it go through chemical reaction, release them at the end of chemical reaction, and then enzyme remains viable and ready for same chemical reaction to be mediated by the same receptor, which comes out of chemical reaction completely unchanged. Some of the proteins are used as the cell identity markers. It is important to understand this concept. It distinguishes our own cells from anybody else's cells unless we have identical twin sibling. Important class of these markers are called major histocompatibility proteins. So the fact that they make our own cells recognizable by our own immune system, being own cells, immune system will not do anything. If the cell would have different major histocompatibility complex proteins exposed externally, that would trigger interesting and swift reaction by our own immune system, trying to eliminate the cell which immune system understands and sees as a foreign content. And finally, some of the proteins that also go through the cellular membrane, they're acting as linkers. They would simply have area that will allow additional structures, in this case, some kind of fiber-like structures, proteins in the form of filaments, to attach to either external or internal surface of the membrane. These filaments will be something that we call also cytoskeleton, although cells don't have a skeleton, but these filaments practically make internal arrangement of the cell, just as our own skeleton would make arrangement for other structures that attach to it, and we see them as muscles or tendons or ligaments that are ultimately forming the shape of our body. So we explained what the concentration gradient is, and we highlighted the role of phospholipid bilayer to maintain it. You realize now any of these ions without integrated proteins that serve the purpose of transportation, it would be remained as a status quo. Nothing will ever change. No particles will reach in the cell. No particles will reach out of the cell. And of course, the cell will be way too isolated to have any kind of chances of survival. Electrical gradient, just comparison between charged particles on two different sides of cellular membrane. Now, from what proteins do, we need to take a look and try to understand how the substances are then transported. What's the driving force between different substances getting in or eventually getting out of the cell? Well, on the previous slide, we talked about the concentration gradient, and we know that when we have particles that are more concentrated on one side than the other side of a medium that we observe, they tend to just spontaneously move until equal concentration is seen throughout. That is what we call diffusion. And diffusion follows its own gradient from high concentration to low. That's how particles move. So, what is required? Some of the particles that eventually are having characteristics that are matching lipids could go and diffuse directly through the cellular membrane. Membrane will not reject them, but they cannot have exceptionally large size. Membrane will not allow it. They cannot have charge. Again, membrane will not allow it. But molecules, for example, of oxygen or carbon dioxide, could easily diffuse from high to low concentration. And it is happening directly. They don't require any specific transportation protein or channel. If the channel is involved, then we understand this substance, whatever gray bubble represents, doesn't have ability to pass directly across the cellular membrane. It would be stopped or banned. So it has to wait for opportune moment and event that would make this protein channel open its pore will allow diffusion from high to low concentration to happen for as long as the protein channel pore is open. When the pore closes, diffusion stops. There is something that is called facilitated diffusion, where the transported molecule cannot be allowed just to diffuse. It requires short-lasting interaction between the protein channel, bonding the transported substance, and then releasing it to the opposite side of the cellular membrane. On an example of this purple hexagonal structure, we can think about, oh, this must be one of the hexose monosugars, whether it's glucose or galactose or fructose. That one will interact with the protein channel, bonding it temporarily before releasing it to the other side of the membrane. And of course, then the cell will receive additional nutrients. You notice that this is all passive transport. And what is meant by word passive? Basically, we say we don't need energy that our cells must consume in order to transport substances. It's going to happen spontaneously on the basis of diffusion gradient. Therefore, no further expenditures of the cell energy is required. But if we want to go against the concentration gradient, let's say this green bubble illustrating sodium, and we want to kick it back from inside of the cell to extracellular fluid where it's already highly concentrated or highly present, then we need to utilize the active transport and use some of the energy that cell has produced, converting adenosine triphosphate into adenosine diphosphate, which will fuel and power up the transport protein to push the transported substance against its concentration gradient and to make sure that this process happens. So diffusion is main stream for allowing substances to go across the cellular membrane for as long as they just go down their own concentration gradient. If we try to reverse it and to go against, then it must be active form of transport. That all applies to particles that are either ions or proteins or carbohydrates or anything else, but not to the molecule of water. And we now have to think about what's happening with the water when it comes to its own interaction with phospholipid bilayer. As we know, phosphate heads, turned both internally and externally, are not going to reject water molecules. They're going to keep them in a close contact with the phosphate heads. Now, the question is, would a single molecule of water be able to move on its own across the cellular membrane? The answer to that question is yes. Why the cell doesn't gain size or shrink on the basis of more or less water molecule movement? Answer is also very simple. At the same time, the same number of molecules of water travel in opposite direction. So for one that comes in, one molecule of water comes out. They don't require protein or any kind of channels because molecules of water are fairly small. They have a relatively large kinetic energy. They move fast and they simply whiz through the cellular membrane, but in a ratio which is 1:1. And that makes content of extracellular fluid, intracellular fluid quite steady without fears that we're going to alter and change the content of intracellular fluid that could be quite detrimental for individual cells. The principle of osmosis actually explains what motivates molecules of water to travel. You can see that in this testing tube, which is open at both ends, we are to pour water, but in the middle of the tube, there is something that is called selectively permeable membrane. A membrane so fine that it would not allow particles of solute, seen on the right side of the tube, to move and follow their own concentration gradient. The membrane will stop them. But the same membrane would not stop movement of water molecules that move from side where solute is less concentrated to the side where solute is more concentrated. So water molecules would move, they would be attracted towards higher concentration of solute. This should be illustrating the beginning of our experiment, and when we allow sufficient amount of time and see that molecules of water have moved across into area where solute is highly concentrated, basically at that point osmosis would stop. For every molecule of water that wants to move from left to the right side of this tube, one molecule of water will go back. It's not because we made concentration of solute same on both sides of the membrane, but rather because of hydrostatic pressure, which built on the right side of the tube, practically the weight of the water column itself is pushing some of it to go back, and then osmotic force requires some molecules of water to keep going back towards higher concentrated solute. And that idea of selectively permeable membrane, practically optimizing both inside and outside of the cell to work like this, is what brings the cell into its own equilibrical state, into its own homeostasis. Should we want to equalize the level of water in both sides of the tube, we would need to apply additional pressure, like some kind of piston, to squeeze it in and to simply force the water to go back across selectively permeable membrane to the left side of the tube. That would restore the starting condition. If we would remove this piston, it will be diagram A. We would be at the beginning, and then massive movement of water molecules would keep moving from left to the right side of the tube until it reaches equilibrium. Water molecules, regardless of their little charge they carry, because they're polarized molecules, hydrogen side being more positive, oxygen side being more negative, as we discussed it earlier, are having pretty much significant kinetic energy, and they can move easily across the cellular membrane. So phospholipid bilayer will not hold them, but rather would not allow any accumulation of water within the membrane itself due to its fatty acid tails and their hydrophobic nature. So let's explain the principle of osmosis by seeing a red blood cell being placed into 3 different fluid-filled compartments. Isotonic solution, which is for human body basically 0.95% solution of table salt in water. Isotonic, iso, the same tonicity concentration of solute. Practically, this would be equivalent of our blood plasma, liquid fraction of our blood, which normally allows blood to move through our blood vessels. And if we place single red blood cell into isotonic solution, so one which is comparable to blood plasma, some molecules of water will come in, but some will be coming out, and a red blood cell will maintain its normal shape. It is going to be seen as some kind of biconcave disc or donut that we can say. But should we change the condition and put same red blood cell into just a regular water, drinking water, without 0.95 addition of salt, that's going to be considered hypotonic solution. And remember, principle of osmosis: low concentration of solute, higher concentration of solute inside the cell. So water will start moving into red blood cell at a larger volume than molecules of water emerging out of it. As a result of more water coming in, this cell will start increasing its size, will look swollen, and at one point it would burst. So that is red blood cell, which has going through hemolysis. It simply bursted due to excessive intake of water into intracellular fluid compartment. However, should we, by mistake, instead of isotonic, make the hypertonic solution, saying 1.5% salt in the water, and then place the red blood cell, there will be a reversed type of osmosis. It's going to be higher solute concentration in a hypertonic solution, forcing or stimulating water to keep coming out of red blood cell. And red blood cell will start shrinking, rapidly losing water. That would be also like the end of the cell. However, the process is called the opposite. It's called the crenation. In medical practice. That's why when a person is suspected to lost significant volume of circulating blood, and when we want to start the IV line, intravenous line, although we may not have blood that immediately could be transfused into the body of a person who extensively bled, it should be the physiologic solution, 0.95% of table salt, sodium chloride, within the water, and that's called a physiological solution that could be easily administered into cardiovascular system, into a vein of a person who needs to receive more fluid in order to help them out to overcome the given condition. Should we opt, there is no physiological fluid, let's use just the water, this would happen to patients' red blood cells. They would keep bursting one after another, so we would create even more aggravated situation. And if we say, well, we don't have it, but we have something that's 1.5%, it wouldn't do really any good to a patient, because red blood cells will undergo crenation. They would start shrinking due to water movement out of red blood cells. So back to channels, back to some kind of new ideas that we want to share. This is channel protein, and this is its pore, and as you can see it is specific for potassium ions. When its gate is open, potassium ions just keep moving from inside of the cell, where they are highly concentrated, to the outside of the cell, where their concentration is low. But when the gate closes, potassium ion movement across the cellular membrane must reach the stop point. So the only thing is, like, then what forces the protein channel to open the gate or to close the gate? These are going to be events that eventually will be part of our studies later on. For example, changing the polarity of the cell. If the event which temporarily disrupts electrical charges across the cellular membrane is an event that could force the gate to open, then we would be talking about electrically stimulating or electrically gated channels. Sometimes the channel might have additional part which acts as a receptor. So binding appropriate ligand to its receptor would make the gate open. Therefore, we're talking about channels that are either electrical or ligand-gated channels. And that will determine their pore being open, allowing the transport, or gate being closed, disallowing the transport across the cellular membrane. Here we have just another example that shows us what's happening with the glucose. We know that the glucose is fairly large molecule, C6H12O6, and as such cannot be transported directly. But of course, its concentration, specifically after a meal, becomes highly present in the extracellular fluid, and it's the cell which needs to get more glucose as the energy to generate more adenosine triphosphate. Of course, the gradient would be high to low, ECF to ICF, extracellular fluid into cytosol or intracellular fluid compartment. And this is transporter protein that we need. As we know, it is going to have a short-lasting interaction, temporarily binding the molecule of glucose and bringing it from extracellular fluid site into intracellular fluid site and releasing it. Now you can ask yourself a question: wouldn't this same protein be able to bind this same molecule of glucose and then bring it back into extracellular fluid? Potentially, yes, but that's why in the biochemistry it's explained. Moment the glucose is brought into intracellular fluid, presence of enzyme hexokinase binds it with a phosphate group. So glucose becomes glucose 6-phosphate. 6 means on the position number 6, where the 6th carbon atom is, it's going to bind a phosphate. So glucose 6-phosphate cannot go back, cannot reverse its journey because addition of phosphate group essentially made the molecule of glucose being arrested within a cell, and it is not having a chance to have its journey reversed back and out into extracellular fluid. Although we know that this is transportation that we consider being active, it is very important. It is so-called the sodium-potassium pump. Extracellular fluid, intracellular fluid compartments, top and the bottom of this diagram. So what's interesting, its proper name is not the pump, it is called sodium-potassium adenosine triphosphatase. It is really this process which is uniquely helping us understand what's going on when we have surplus of sodium within a cell and relative lack of potassium within a cell. For every 3 ions of sodium that would be exported out of the cell, the same pump, sodium-potassium pump, after it invested some energy using adenosine triphosphate and converting it into adenosine diphosphate by binding the phosphate group, basically brings additional 2 potassium ions back into the cell. And then the pump releases the phosphate group because that was the energy that was required. So through this example, we understand what is the transport against the concentration gradient, and it goes in an odd ratio. For every 3 sodium ions, basically 2 potassium ions are imported into the cell. Sodium is exported against its own concentration gradient into ECF, where it's already present in a high concentration, and potassium also becomes imported against its concentration gradient, because we already know potassium is the major intracellular positively charged ion. So this is example of active transport, which really handles 2 most abundant ions, sodium and potassium. This is going to make things just a little bit more kind of interesting. There are some protein channels that are called antiporters, and antiporters means within the same cycle of this enzymatic pump. Basically, we have 2 substances that are moved across the cellular membrane, but it goes in opposite direction. Sodium is brought into the cell, but calcium ions are exported, or we see here sodium being imported and exportation affected hydrogen ions. We know that sodium gradient is easily followed, so high to low, and this is what energizes the pump. It is movement of sodium particles that really help us expel calcium or hydrogen. Or it could be a symporter type of thing. Again, not using adenosine triphosphate, but we see that a long single sodium ion, basically molecule of glucose, could be imported, or amino acid could be imported from extracellular to intracellular fluid. In this case, we're talking about symporters because substances travel in the same direction. Antiporters are protein channels that allow one substance to travel in while the other one travels out of the cell. We're almost towards the end of this presentation about cellular membrane, and just we need to address what's happening with the larger substances, something that simply cannot utilize a protein. How do we then get them in or out of the cell? In this case, we're talking about transportation that is endo or exocytosis, endo in, exo out of the cell. So let's see here what's happening through this short but interesting example of low density lipoprotein and its ability to bind on its own receptor that the cell has on its surface. Cells certainly would have more than one receptor, so it will bind multiple low density lipoproteins. but not throughout the cellular membrane, only at one fragment of the membrane, which on the underside of a cell membrane has clathrin-coated area. That's another protein which is manufactured by cell. So when all receptors are bonding low-density lipoprotein particles, cellular membrane in the area which is clathrin-coated starts invaginating, creating a little pit, and that pit will progressively become deeper until all receptors are saturated, and then the bubble will pinch itself off from a cellular membrane and travel into cytosol. Moment it is inside the cytosol, clathrin protein starts uncoating the transportation bubble or vesicle, and it goes back to a different area of the cell where it can be reused for this process over and over. In the meantime, the uncoated vesicle without clathrin molecules fuses with other cellular organelle called the endosome. As it fuses with the endosome, it releases the substance that has been transported. That's the low-density lipoprotein. And you can see here that the endosome actually looks like creating those vesicles on 4 different ends of the molecule. These 2 ends that look like just little 2 handles are obviously to pinch off and to travel back into direction of cellular membrane, because they would still have receptors that are now to be ready for another cycle, while the part of the endosome, which is just containing transported molecules of low-density lipoprotein, essentially pinches itself off and travels until it merges with a lysosome organelle that will contain a number of different enzymes that'll start further splitting low-density lipoprotein and practically break it down to substances that are either to be directly used for cells metabolism, or they will be recycled in the form of synthesis that the cell is to do, or simply would be metabolized and its residual products will be eliminated from the cell. That is example of endocytosis and transportation vesicles. It's always the same principle that part of the cytoplasmic membrane pinches itself off, making a vesicle, a bubble, that transports the substance without interacting with the rest of the cellular cytosol. If we take a look at what's going to happen to for example, to microorganism, which also has been linked to some of the receptors on the surface of the cell's going to respond in the same manner. It will create endocytosis. It'll form the transportation bubble that will be called the phagosome. We imported the microorganism into the cell, but then the phagosome will bind to lysosome, which again will release a number of its enzymes that will simply affect the imported substance, microorganism, breaking it apart, and that will prevent microorganism causing any kind of serious damage within a cell. Our immune system and some cells work exactly like this. In the case that we have fluid, different type than just water, we're talking about cellular drinking, not cellular eating, and that's why it's not called endocytosis, but rather pinocytosis, process that involves formation of pinched off segment of a membrane, forming a transportation vesicle, bringing up fluids that would be again used by the cell after the transportation vesicle binds to lysosome, which, due to its number of different enzymes, will interact with substance being transported and, of course, will trigger either preferred reaction by the cell or will have a different destiny depending on what is transported into the cell. At this point, we would start looking into a number of different structures inside the cell, and this slide illustrating microfilaments, intermediate size filaments, or microtubules will be the first slide for the next segment of this video presentation. We would stop here for a moment and we'll continue with microfilaments, microtubules, intermediate filaments to continue looking into cytosol and to say a few words about other intracellular organelles.